You know, for over a century, the lab mouse has been our single greatest ally in the fight against human disease. From cancer to immunology, pretty much everything we know we owe to them. But what if I told you that for decades, we've been building this incredible foundation of knowledge on a dangerously incomplete blueprint? It all boils down to one kind of shocking question. I mean, really think about that. Why would we do that? Imagine all the incredible diversity of people in the world. Now, what if we based almost all of our medical knowledge on just one person? It sounds absurd, right? Well, that is exactly what we've been doing with mice. And it's created a massive, massive problem. And this is the perfect way to picture it. If you have a map of Paris, you're going to be great at getting around Paris, no problem. But the second you want to drive to Berlin or Rome or anywhere else, you're completely lost. Worse than that, you wouldn't even know what you were missing. And for decades in genetics, we've been stuck in Paris. So this huge problem actually has a name. It's called reference bias. Ever since 2002, the official map for the entire mouse genome has been based on one single strain. It's called C57BL6J, or just BLA6 for short. And by relying only on this one map, we've been totally blind to the vast genetic landscapes in all the other mouse strains. Landscapes that, it turns out, hold critical secrets to fighting disease. And this isn't some small academic issue. The problem has absolutely exploded. Just look at this. Back in the 80s, about a quarter of studies used this one mouse strain. But today, that number has shot up to over 70%. The very existence of a single, easy-to-use reference map created this feedback loop, where it was just easier for scientists to use the one mouse we had a map for. Okay, so how do we fix this? How do we break out of this cycle? How do we finally create a map of all of Europe, not just Paris? Well, a team of scientists decided they had to build something completely new, a mouse pangenome. So instead of just sticking with that one mouse, they sequenced the complete genomes of 17 different strains. And you can see the amazing diversity right here. They didn't just include 12 of the common lab strains, they also, and this is so important, included 5 strains that came directly from wild mice. This captures a staggering amount of genetic diversity that we've been totally ignoring until now. A pangenome isn't just another map, it's a full-on atlas. Think about it this way. It still has that main map, the BL6 genome, as a kind of backbone. But then it adds in all the alternate routes, all the unique landmarks, all the hidden regions from the other 16 genomes. It's literally the difference between a folded paper map and Google Earth. So with this incredible new genetic atlas in hand, researchers could suddenly see things that were hidden in plain sight the entire time. And their first discoveries were in one of the most important areas you could possibly imagine. The first huge breakthrough came when they looked at one of the most important and, honestly, one of the most notoriously complex parts of the genome, the region that controls our immune system. And this shows you exactly what they found. There's this family of genes called H2, and it's essential for your body to tell what's a friend and what's an invader. On the old map, it was a fragmented, tangled, confusing mess. But just look at how the pangenome fixes it. That jumbled knot is finally assembled into a clear, continuous line, showing us its true structure for the very first time. But it gets even crazier. It turns out it wasn't just that parts of the old map were blurry. No, entire sections were just missing, gone. The pangenome let researchers go on a kind of genetic treasure hunt for genes we knew had to exist, but we could never, ever find. And guess what? They found them. A stunning 278 gene markers that were just completely absent from the official reference genome were finally located. These were genes that had been discovered and studied years, sometimes decades ago, but were basically lost to science because they didn't exist in that one BL6 mouse. A perfect example is a gene called DFO1. It's part of a whole family of genes that are crucial for shaping our gut microbiome and fighting off infections. Scientists had actually cloned it from other mice years ago, but when they looked for it on the official BL6 map, it was nowhere to be found. Now, thanks to the pangenome, we know exactly where it is and how it fits into the bigger picture. This new atlas also let us see a really mysterious type of genetic variation called a VNTR. Now, you can think of these as little genetic stutters, where a sequence of DNA just repeats itself over and over and over. They were called hidden for a reason. Our older technology just couldn't read these super repetitive regions accurately, making them another huge blind spot. So how big was this blind spot? It's kind of shocking. Researchers found these complex, stuttering structures inside 428 different genes that make proteins. That is almost 3% of all the genes in the mouse, and a lot of them have been linked to human diseases. This was a completely unexplored frontier of genetic variation. Okay, so we found all this new stuff. It's fascinating, right? But what does it actually mean for the real world? How does a better map actually lead to better medicine? Well, it turns out that having the right map changes, well, pretty much everything. Because if you're using the wrong map to analyze your scientific data, you are absolutely going to get the wrong results. And this is especially true for one of the most common techniques in all of modern biology, something called RNA sequencing. This slide just says it all. Look at the left. That's what happens when you analyze genetic data using that old single map. See how the peaks in the graph are all messy and the alignments are scattered? It's just noisy and inaccurate. Now look at the right. That's the exact same data analyzed using the correct map from the Pangenome. The signal is crystal clear, the peaks are sharp, the alignments are perfect. This one change improved mapping accuracy by over 5% and totally changed the results for over 2% of genes. And that kind of accuracy, well, that's what leads to the ultimate payoff, solving real medical mysteries. Let's look at an absolutely powerful example involving one of the world's deadliest diseases, tuberculosis. So scientists knew that some strains of mice were highly resistant to TB, while others weren't. They just didn't know why. So using a diverse group of mice, they went hunting for the genetic reason. And this graph, this became their treasure map. Do you see that huge peak? That massive spike on chromosome 15 was basically screaming, the answer is here. It pointed them to the exact spot that controlled resistance. So what was in that spot? The Pangenome gave them the answer. The resistant mice had a whole group of special immunity genes called apolipoprotein L genes. And here's the real kicker. These life-saving genes were completely absent from the old BL6 reference mouse. The cure was literally hidden in a place our old map couldn't even see. This is a total game changer for mouse research, obviously. But honestly, this whole story is so much bigger than just mice. This revolution in how we see a genome is changing the future of medicine itself. You know, the authors of the study really summed it up perfectly. For decades, we have been working with a built-in bias, totally blind to crucial pieces of the puzzle. This mouse pangenome has given us a new set of eyes, allowing us to see the full, complex, and beautiful picture of genetic diversity for the first time. And that brings us to the final, and let's be honest, the most exciting question of all. We are right now building a pangenome for humans, one that represents the incredible diversity of people from all around the world. So if switching from one simple map to a full genetic atlas revealed this much about disease in mice, what earth-shattering secrets about our own health are we about to uncover?